suspension, and a colloid. The solution is homogeneous. It would be like bottled water because they all taste different. So they all have different minerals, but you don't see them. It is a homogeneous solution. Solutions are homogeneous. Suspensions are heterogeneous because once that blood sample sits, the cells settle to the bottom. And then a colloid is also heterogeneous because it's a cell. It's a cell with all of the organelles. Know the organs in each of the 9 abdominal pelvic regions. I may say that the stomach, the gallbladder is contralateral to the stomach. So you have the stomach in the epigastric region, and you'll have the gallbladder in the right hypochondriac region. So it would be opposite, not ipsilateral, which will be the same side. The urinary bladder is hypogastric. So know those 9 or abdominal pelvic regions. And I actually keyed it into what we see often in the ER: appendicitis, splenectomy, damage to the spleen, liver issues, fatty liver, hepatic issues, which hepatic means liver. Also stomach ulcers, appendicitis. So those that were in the lab, and I'm going to go through the lab with you. But that will help you just narrow down, because there are a lot of organs in everything in the abdominal pelvic region is in those 9 abdominal pelvic regions. But we'll key it down to just those that we see often. Know the function of each of the organs covered in class. This is typically a matching question. And I have a diagram for you with all 11 organ systems, the function of that organ system, and the organs in that organ system, and it will be major organs. And we'll go through that when we review lab exam 1, but it is in chapter 1. Know the tissue types and a representative organ that will contain each tissue type. The way I will teach you that tomorrow is that we have 4 types of tissue: epithelial, connective, muscle, and nerve. Of those 4 tissue types, there are 8 types of epithelial, and we'll go through those. And one of my short answer questions could be, name the 8 types of epithelial tissue. And those 8 types of epithelial tissue, it will come up later in this study guide, but I'll give them to you now. We either have 1 layer or more than 1 layer. 1 layer is simple, more than 1 layer is stratified. And then we have the shape of the cell, the shape of the cells in those tissues. They can either be flat, and if it's 1 layer, that's simple squamous. If it's cube shaped, simple cuboidal, or if it's tall, simple columnar. And then for stratified, we have stratified squamous, cuboidal, and columnar. That's 6. And I said there are 8 types of epithelial tissue. Pseudostratified, ciliated, columnar epithelium. Pseudo means false, pseudopods, false. So pseudostratified means it appears to be stratified, but it's not. And then I put the cilia in there to help us learn that tissue. And then there's transitional. So know the tissue types and a representative organ. Simple squamous, and the way I'll teach it, and when I go through it with you in a minute for lab, which is the same as in my lectures, I give you a specific sample because structure is complementary to function. The lungs, in the alveoli, the air sacs in the lungs, we exhale CO2 and bring in oxygen. So I want 1 layer and I want them flat because I want that quick gas exchange. So simple squamous is in the alveoli, the sacs of the lungs. Then I have simple cuboidal. That will be in the kidney tubules. Okay, I have uric acid, other waste products, but I have to keep the sodium, all the electrolytes, keep the water. So the kidneys have to have time to filter. So I don't want them to be flat, but I don't need a whole lot of time. So that is simple cuboidal, is a type of tissue found in the kidney tubules. And now I go to the intestines. I have eaten a pretty good dinner. So now I need to absorb all those nutrients into the bloodstream. So I need some time. So that would be simple columnar, tall, simple columnar in the intestines. And then stratified squamous, because our skin is layers upon layers of cells. When we get to chapter 5, I'll talk about the top layer sloughing off. So it is stratified squamous in the skin. And then the other two types of stratified, I don't teach. I teach that they exist because the apical layer is how we name them, but I don't use examples for those in the body. So that's only of the 6, that's only 4 that I've talked about. The other 2, transitional, is in the bladder, the urinary bladder. Transitional means it looks like it's tall, then it looks like it may be flat. It tends to be transitioning based on the fullness of the urinary bladder. So an example, in the bladder, in the urinary bladder, an example for the bladder would be transitional. And then we have pseudo stratified ciliated columnar. That's in the trachea. When we breathe in air, if there's a dust storm, some of the hairs in the nasal cavity might trap some of the particles, but we don't want anything getting deep into the lungs. So the pseudo stratified ciliated is in the trachea, spreading mucus to trap debris to keep it from getting deep in the lungs. That's why I said the ciliated. We have ciliated and unciliated, but I teach that so I can help my students just starting learn. So the tissue types and the representative organ I just gave you, and I'll repeat it in class tomorrow for the simple: squamous, cuboidal, and columnar, stratified squamous, transitional, and pseudostratified. Just gave you the representative organ and the type of tissue in each one. The types of cellular transport mechanisms, I think you've got this because this is SLOD. We talked about passive transport, which either needs a carrier or a channel. Remember, we even need an aquaphore channel to get water in and out of the cell because of the lipids inside the cell membrane itself. So to get through that membrane for water, to get through the oil, we need a channel. So we either have channel or carrier mediated, passive. Osmosis is the diffusion of water across a selectively permeable membrane. That's passive. And then for active primary, I use the sodium potassium pump, because sodium, after it rushes in to cause an action potential, then it's going to be actively pumped back out, which is where most of it is. We need ATP to do that. But then we have secondary active transport. It can either come in with sodium or leave out with sodium. And I usually use glucose as an example. It's the gradient of sodium that drives the transport of glucose secondarily. So that's secondary active transport. And then vesicular endocytosis, exocytosis, transcytosis also requires ATP. It is active transport. I also talked about the passive mechanisms for transport. I did those first. Simple diffusion. Spray perfume in the front, you move it in the back. Gases can go across that membrane freely, simple diffusion. And then I already mentioned channel or carrier mediated or facilitated diffusion, passive. Where in the body would you find each type of cartilage? I talked about the 8 types of epithelial tissue. Cartilage is a connective tissue. We have 3 types of cartilage, which is connective tissue. We have adipose, which is the most, time out, I can't take those words back. Cartilage, 3 types of cartilage. We have fibrocartilage, elastic cartilage, and hyaline cartilage. Hyaline is the most numerous. It connects the ribs to the sternum. It's on the tip of the nose. It's on the end of the long bones. That's the most numerous, hyaline cartilage. And that one, I'll show you a photomicrograph. It will be on the exam. Then we have elastic cartilage in the earlobes and in the epiglottis. And then we have fibrocartilage. It's between the vertebrae, in the knee, where the femur sits on top of the tibia, and it's in the pelvic bone, pubic symphysis. It's in the pelvic bone connecting our 2 pelvises together. So fibrocartilage, elastic cartilage, and hyaline cartilage, 3 types of cartilage. I mentioned adipose, that one will be on the exam. It's a type of connective tissue, cushioning. Then we have connective tissue proper. We have bone and blood, which are also connective tissues, and then the 3 types of muscle. Know an example of an organ that is exocrine, endocrine, both, holocrine, merocrine. This one will also come up later on the exam. There's often not just one question. There can be multiple questions on the same concept. Exocrine glands, exocytose, they excrete. Endocrine refers to the endocrine system, which is the endocrine, which means it secretes into the circulatory system. So that is the endocrine system. It secretes into the circulatory system. So endocrine secretes inside the circulatory system. Exocrine excretes to the outside. 3 types of exocrine glands. We'll finish chapter 3 tomorrow. The 3 types of exocrine glands. Merocrine. Merocrine type of secretion, which means exocytosis onto the surface. And the merocrine, an example for that type of secretion would be the sweat glands. Then you have apocrine secretion, which I don't have typed here. That is a second type of exocrine gland. And an example of that are the mammary glands. The apex of the cell ruptures and it exocytosis. Mammary secretions, mammary glands, apocrine. Then the 3rd type is holocrine. That is the sebaceous oil glands. The sacs with the oil rupture. So it's a holocrine secretion, and those are the sebaceous oil glands. For both, an example of an endocrine inside the bloodstream, exocrine exocytosis, would be the pancreas. It is both exocrine and endocrine. Exocrine because it sits right under the stomach, and it exocytoses digestive enzymes into the stomach. That's that exocrine methodology for the pancreas. Endocrine for the pancreas, it secretes insulin inside our bloodstream so that we can reduce the glucose in our blood. So pancreas is endocrine with insulin, exocrine with digestive enzymes. So it is both endocrine and exocrine. What type of tissue is found in the skin? I've already said an example is stratified squamous epithelium. Know the process of transcription, translation by definition, and where the process occurs. This is the central dogma. It's taught in general bio, gen bio, all types of biology, even to be in high school. So I'm hoping this is a review to get you points on the exam. By way of review and where each occurs. So I will explain. The DNA is in the nucleus. It never leaves the nucleus. The DNA is like the cookbook for anything we ever want to cook. Any protein in our body we want to make, the recipe is in the DNA in the nucleus. So how do I know how to make it? Well, it is transcribed. The genetic code is transcribed from double-stranded DNA in the nucleus to single-stranded. And this is another question on the exam somewhere. Name the 3 types of mRNA. I'm about to use them in my example. So the double-stranded DNA in the nucleus is transcribed or rewritten onto single-stranded messenger RNA. The single-stranded messenger RNA leaves the nucleus and sits on the ribosomes outside the nucleus. And the ribosomes have my second type of RNA, ribosomal RNA. And now the message in the messenger RNA, which has the codon, is translated into protein with the help of transfer RNA, which has the anticodon to match the codon on the messenger RNA. The translation is with tRNA, bringing in the corresponding amino acid on that growing polypeptide to form the protein. And that occurs outside of the nucleus, so it's in the cytosol. So you have transcription in the nucleus from DNA to messenger RNA. You have translation inside the cytosol with messenger RNA sitting on the ribosomal RNA in the ribosomes and being translated to protein with the help of tRNA, bringing in the specific amino acid. Know the organs in each of the cavities discussed within the ventral and dorsal body cavity. I'll do a picture review just a minute because lab reinforces lecture. So I'll run through the labs for pictorial view. Okay, I was looking at some chats. Hopefully you're answering each other's chat. So each of the cavities discussed in the ventral body cavity, we have the brain and the spinal cord, ventral and dorsal. In the dorsal, PD, posterior dorsal. In the dorsal body cavity, we have the brain and the spinal cord. In the ventral, AV, anterior ventral. In the ventral body cavity, we have the thoracic cavity and the abdominal pelvic cavity. In the thoracic cavity, and this is going to come up in a little bit, I'll cover it now. You have the heart and the lungs. The heart is in a pericardial cavity, and it is covered by serosa, a membrane to keep it from drying out. The serous membranes, there are two: parietal for the cavity, visceral for the organ. So if I say the patient had fluid within the heart, it would be in the visceral pericardial cirrhosis. If I said the patient had fluid around the heart, then it will be in the parietal pericardial cavity. Plural, both parietal and visceral, are for the lungs, and peritoneal for the intestines, both parietal and visceral. So the cavities, the thoracic and abdominal pelvic, the ventral, thoracic and abdominal pelvic, dorsal, the brain and vertebral. Positive and negative homeostatic feedback loops. There are short answer questions on the exam too. The short answer, one of the short answer questions could be explain positive and negative feedback loops. The way we learned it is that a positive feedback loop, if the body is generating a positive feedback loop, it wants you to continue in the same direction, more of the same. If the body is generating a negative feedback loop, it wants to reverse the stimulus. If I am cold, the body says shiver to warm my body temperature up. If I'm hot, the body says perspire to bring down the temperature. The two positives I taught are blood clotting, because you want more and more platelets, and childbirth, because you want more and more contractions to birth the child. But they won't be that clear cut on exams. It will say the patient ran through poison ivy and just began to itch and itch and itch. What type of feedback loop? And it's more and more itching, so it's a positive feedback loop. Or the patient has a tumor that is causing an increased or continuous production of TSH. Continuous, that's a positive loop, more of the same. The body is telling it to make more and more. So just read it: positive, same direction; negative, reverses the direction. The cell model will be on there. You'll need to know the nucleolus, and I don't take off the spelling, but you've got to prove to me you're saying nucleolus and not nucleus, because I need to see that O. The nucleolus is where RNA is made. The nucleus is where DNA is stored. Then you have peroxisomes, peroxosomes, oxidize, so they're going to break down compounds in the liver. We have lysosomes, which lyse. We have the mitochondria, production of ATP. We have smooth endoplasmic reticulum, because the cells are 60–80% water, and a lot of our hormones are lipids. So we're going to need somewhere to store them. That will be in the smooth ER. You have the rough ER. It's rough because it's studded with ribosomes. It's typically right outside the nucleus. And then you have the Golgi apparatus, and those can make vesicles for transcytosis and other activities within the cell. So we'll go through the cell model. There will be a picture of the cell, name these organelles A, B, C, or D. Know the muscle types where they can be found in the human body. 3 types of muscle. Skeletal, which is on bones, skeletal muscle. Smooth muscle is any muscle that we don't control. So in the intestines, in the lungs, in the bladder. And cardiac, it's also involuntary, but it is only in the heart. Blood vessels have smooth muscle. So it is only in the heart. So you have cardiac muscle in the heart, involuntary and striated. We have smooth muscle. It is not striated and it is also involuntary. And then we have skeletal muscle, voluntary and striated. And there'll be pictures of those on the exam, but I will show you that in a minute. Anabolic and catabolic reactions. Anabolic, dehydration synthesis. Catabolic, hydrolysis, breakdown. We went through the 3 serous membranes, parietal, pericardial. See, that's not right, not parietal. Pericardial, pleural, and peritoneal. And within those 3 serous membranes, it could either be lining the cavity, that would be parietal, or lining the organ in that cavity, and that would be visceral. What are chemical reactions? It is when substrates come together to form products, or reactants come together to form products. Where do you find each type of tissue we studied? I went through that already, and I'll go through it in a little bit here again, but I talked about that. Apoptosis. A red blood cell has a definitive lifetime, after which apoptosis is just automatically degrades, and the heme portion of that red blood cell is recycled to make a new red blood cell. So it is not abnormal. It is normal for our cells to apoptose, just degrade, where new cells are made to carry out their function more precisely. Homeostasis is trying to maintain stable conditions within the body, returning the body to its normal state. What are solvents? It is usually in the greatest proportion, it dissolves the solute. We did the one in the lab where it was crystals on a solid agar, so both were solid. The crystals in the smallest amount were the solute. The solvent in the greatest amount was the solid agar plate. So the solvents dissolve the solutes. The pH of human blood is slightly basic, 7.4. And I talked about the only voluntary muscle is skeletal, cardiac, and smooth are involuntary. Be able to correctly use the directional terms. So I would review proximal and distal. Proximal is coming in toward the body. The elbow is proximal to the wrist. Distal is going away, distant from the body. The elbow is proximal to the wrist. The wrist is distal to the elbow. Superior, inferior, etc. So review those. I'm going to run through them here with you in a minute. The 4 chemical elements found in the human body: carbon, hydrogen, and oxygen. And nitrogen is the 4th one. And nitrogen is found in proteins and nucleic acids, DNA and RNA. Again, know the function of the cellular organelles. I reviewed that with you. I am continuing because I haven't covered most of this. Are y'all with me? Are we good?
Yes.
Okay, good.
Yes,
I
we're
appreciate
good.
the feedback. Okay, am I going too fast?
Yes.
Yes.
Please stop me if you don't get something. This is a new approach of going through it before I finish all the material. So now tomorrow, our last chapter, 3 and 4, you'll know what to be looking for. So you're actually on a mission to make sure to reinforce for the exam. So I'll try to slow down a little bit. Any questions so far? Anything you didn't get that you want to ask me before I continue? No, we're good. I know somebody wants to say something, but who wants to be the first one? We're good? Keep going?
Yes, we're good.
Okay. Thank you. Okay. So the chemical elements, carbon, oxygen, hydrogen, and nitrogen. The function of cellular organelles discussed, and I just went through that with the peroxisomes and lysosomes, nucleolus, nucleus, mitochondria, Golgi, smooth and rough ER. The three types of RNA were the messenger RNA, carries the codon, transcription of DNA. The ribosomal RNA is in the ribosomes, and the transfer RNA brings the specific amino acids corresponding to the codon for the growing polypeptide, which will be my protein. And the tRNA has the anticodon. Know the organs within each organ system. This is another matching. You'll match the organ systems with and the function of the organ systems. And when I go through the diagram that I have in your lab to study, I'll point that out. Survival, I pointed that out in lecture. We don't have to reproduce to survive. We need proper atmospheric pressure, water, proper temperature. But reproduction and growth are not survival. They're indications of life, but not survival. What are solvents again? It's another question. How is DNA replicated? And it is semi-conservative. DNA is double-stranded, and the way it is replicated, the helix is unwound, and then we make a new strand complementary to each old strand. So it is semi-conservative because there's one old and one new strand. Know the regional terms and how to apply them. We just went through that. I'll go through it when I review the lab. Mitosis. By function and end result, if I start with 8 chromosomes, after mitosis, I am going to end up with 8 chromosomes. MEIO, meiosis is reduction division, and you'll get that in AMP2, the reproductive system. Mitosis is just straight duplication. What I've got here, if I duplicate it, I'm going to have the same thing here. 8 chromosomes here. Know the cellular membrane structure. We went through that. It is, you have your phosphate heads toward the outside and inside of the cell. And on the inside of that membrane, those lipid tails are toward each other. So the inside of the membrane is hydrophobic, but the inside of the cell and the outside of the cell is hydrophilic because we're 60 to 80% water. But the cellular membrane is a phospholipid bilayer. Lipid tails turned in toward each other. Know the body planes and how to apply them for a surgical procedure relative to the human body. To amputate an arm, parasagittal. To amputate a toe, frontal. To amputate a leg, transverse. And don't forget, if I say I carve something, a Thanksgiving turkey, into equal left and right halves, and ask you if that's a sagittal cut. Well, if I have median there as opposed to just sagittal, median is a more correct answer because the sagittal could either be para or mid-sagittal. Mid-sagittal is equal left and right halves. If mid-sagittal is not there and median right along the midline, that would be a better answer. Isotonic, hypotonic, and hypertonic for osmotic concentrations. I'll show you that when we get to the lab, and we'll finish chapter 3 tomorrow. Isotonic is the same, which is 0.9% sodium chloride. Hypo is low. My example there is distilled water and the cells rupture. Hyper is more. My example there is anything greater than 0.9. I usually go 10% sodium chloride, but 5% would be hypertonic. Know the cell junctions. We talked about those on last week. Desmosomes hold cells together. Tight junctions seal, and gap junctions allow for communication. Know that a cell is the smallest unit of life. That's the answer. What's the smallest unit of life? A cell. Be able to list, I did this already, the three classes, and read the questions. Sometimes they have two parts and my students only answer one. Be able to list the three functional classifications of exocrine glands. So now I want the functional classification. Based on how they secrete, that is the functional classification. It tells you how they secrete. Additionally, provide an example of each type of exocrine gland while listing its mode of secretion. And I said there are three functional classifications, and I've already told you merocrine is the functional classification, and an example would be the sweat glands. Apocrine is a functional classification. An example are the mammary glands. Holocrine is a functional classification. An example are the sebaceous oil glands. Know the cell cycle. We didn't quite get there. I will get there tomorrow. The cell spends most of its life in interphase. It's just sitting there growing, reproducing, and then when it gets ready to divide, it's going to duplicate the DNA, duplicate the organelles, then it's going to go through division, which is mitosis, PMAT, prophase, metaphase, anaphase, telophase. And then it will cytokinase, break into two distinct daughter cells. So the cell cycle, I'll show you that in a minute. It's in one of the labs you'll do tomorrow, and it's in my lecture tomorrow. Know the functions of DNA, which carries the genetic code, and RNA. It helps with the replicative processes and the three types of RNA. Messenger RNA carries the codon. It's a transcript of what's in the DNA. Ribosomal RNA is in the ribosomes, and transfer RNA brings in the amino acid for translation. For the lab exam, what happened in lab exercise 2 with respect to adding acid to buffers into water? Just read the question. Sometimes, and we're human, I do it too, we'll read the question knowing what the answer is, but you have to force yourself to slow down and read the question as written. When we added acid to water, the pH drastically decreased. When we added acid to the buffer, the pH slightly decreased. Also, CO2 was liberated when we added the acid to the buffer. Know the process of mitosis and the end product of a cell that's completed mitosis. If I start with 8 chromosomes or 4 chromosomes, whatever I start with, that's what I end with after mitosis. Know the directional terms and how to use them. Review lab exercise 1. I will do that in a minute. Know the planes of the body and how to use them. I will do that in a minute. I already talked about the mid-sagittal, parasagittal, frontal, and transverse. Know what happens to RBCs in hypo, hyper, and isotonic solutions. I will show you that in the lab, because what I'll do is just put A, B, and C on a picture, and I'll say, based on what happened to the red cell, what type of solution was it placed in? If it burst, it was hypo. If it crenated or shrank, if it has the cell crenated or if it shrunk, then when the cell shrinks, which is crenation, that is hyper. And then iso, nothing happens. It's an equal concentration. Know the organs in the 9 abdominal pelvic regions. We'll go through lab 1. Which is where we pointed out just those common things we see. Significance of nitrogen in the body. Nitrogen is found. It's one of the 4 macro elements found: carbon, oxygen, hydrogen, and nitrogen. The nitrogen is found in proteins and nucleic acids. The function of the organ systems I covered in I said I was going to show you. Like the respiratory system, integumentary system will be hair, skin, and nails. It protects. But I will show you the one page that I have for learning the 11 organ systems. Quick acting would be nervous system. Muscular allows us to manipulate our environment, but we'll go out and look at that handout. Now the substances, know how substances cross cell membranes, know and understand and be able to critically apply to a situation. And I define passive transport, active transport, vesicular, primary and secondary active, those concepts will all be on there. Passive diffusion, and then osmosis, the passive diffusion of water across a selectively permeable membrane. Be able to recognize the various phases of mitosis. I will show you that in a minute. I usually stick to two. I said the four are, there's actually more than four, but in the simplest of terms, prophase, metaphase, anaphase, and telophase. I stick to metaphase and anaphase. Metaphase is directly on the center of the cell, the equator, and then anaphase, they're beginning to be pulled apart. I will show you that. You'll look at slides tomorrow. Know our state of hydration and that the sodium potassium pump must be in homeostasis to ensure the body performs normally. Yes, you can drink too much water. It is called hyponatremia. I think that's what this is getting at. Anabolism, dehydration synthesis, catabolism breakdown. The serous membranes, we've talked about them, pleural, parietal, pericardial. I keep saying parietal. Pleural, pericardial, peritoneal. And within those three, it can either be parietal for the cavity or visceral. So pleural for the lungs, pericardial for the heart, and peritoneal for the abdominal pelvic cavity, specifically around the intestines. Know how the urinary and respiratory systems work to help us maintain acid-base balance in the body. So the question may describe a patient that is in metabolic alkalosis or acidosis, and it may say what organ system is a quick-acting organ system to help that patient return to homeostasis. It would be quick-acting, it would be the respiratory. Because if we're in metabolic acidosis, I need to lower my pH because I'm in acidosis. I want to come to slightly basic. So therefore, I am going to hyperventilate, release more CO2 so that I don't have the CO2 to combine with water to form carbonic acid. However, if I'm in metabolic alkalosis, pH is too high. I want to bring it down, then I want to slow my respiration to hold some of the CO2 to make carbonic acid to bring my pH down closer to 7.4. Urinary system will also eventually, I just ate a whole case of oranges, citric acid. So my pH is too low. So the urinary system will eventually, it'll take a time, but it'll eventually excrete those products that are affecting the acid-base balance in the body. So these are the two organ systems that help us maintain acid-base balance in the body. Know how substances cross a selected permeable membrane to achieve equilibrium. That's exercise 6. I'll go through that in a minute. Regional and directional terms in lab 1 and chapter 1 and be able to correctly use them. The pH of blood is slightly basic. We'll do the microscope lab tomorrow. We have parfocal scopes. You focus them on scanning power, which is 4, and the magnification of looking through that scanning objective. The ocular gives you 10, so 10 times 4 is a 40x magnification. We'll go through that tomorrow. And it's parfocal because once that object is, I'm going to scan that slide, patient is thought to have malaria, I'm going to look at the entire slide. And then if I think there's a malaria parasite, I'm going to go up to low or high power. I'm sorry, I'm looking at the chat that I am going too fast. I think Karen said she had recorded it. So if y'all can share with each other those in the chat, but I am hoping that you will go through this on your own and then don't hesitate to text me. Hey, this question, I can't figure out the answer. What does it talk? I'm happy to correspond with you, but you haven't seen this material. It's kind of different approach for me to do the study before I finished lecturing on chapter 3 and 4. So now you can key in onto what you should be gleaning from 3 and 4 as you learn the material, because I know a lot of times my students feel overwhelmed, especially if they're new to A&P. So give me feedback, and it's already 9:50. I've had you 50 minutes. It's going to take a few more minutes because I want to show you photomicrographs. Melissa, talk to me. Because I saw your chat. I know it's going fast. I don't want to pick on her. There's several people that said it's going fast. And Karen says she would share the video. So tomorrow in class, bring your computers or devices or emails in. It's going to be big, which is why I don't record it. It'll take up a lot of space. So you may want to bring thumb drives so you can download it and take it home if you want a copy. Is that okay, Karen? And who else is recording? Sophia. Who are my other people recording?
I'm recording.
Madeline. Thanks, Madeline. And who else?
I think I might be recording, but I'm not sure.
I'm recording, Kiara.
You are, Kiara. Thank you. So Kiara, Karen, and Madeline. And Rosalind, I thought I gave permission. Are you?
I believe I am. I think I am. There's like
Okay, so there are 4 people that are recording. Melissa, did you not engage recording? Okay, the microscope. Know the organ systems and the organs in each system. I will show you a handout for that. The cell and be able to recognize the parts. We talked about that in lecture. Explain positive and negative feedback and provide an example for each. Sounds like a short answer, but we talked about that with lecture because lab reinforces lecture. Know the parts of the cell. So here there could probably be a diagram either here or on lecture. And I'll say, what is this organelle, this organelle, this organelle? Know the difference between prokaryotic and eukaryotic. That will be the lab you'll do tomorrow. Prokaryotes, for those that have had micro. Pro is before the karyote, which is the nucleus. So prokaryotes have no nucleus. That's like bacteria. So they have, in fact, no membrane-bound organelles. And eukaryotes have membrane-bound organelles. The 8 types of epithelial tissue, I already did those for you. I said the simple and the stratified. 1 layer or more than 1 layer. 3 types of simple based on cell shape: simple squamous, simple cuboidal, simple columnar. Same with stratified, more than 1 layer: stratified squamous, stratified cuboidal, stratified columnar. That's 6. Number 7 is transitional, like in the bladder, and number 8 is pseudo stratified, ciliated columnar epithelium in the trachea, keeping dirt out of the lungs. The cell spends most of its life in interphase, and then it will eventually divide. So it'll go through the M cycle, which is mitosis, PMAT, and then it will cytokinase. Know the tissues. You will be given a photomicrograph of the tissues we studied, and you will be asked to name this tissue and be specific. There will be no word bank. I will go through, because here are the tissues that you'll have to name. So I am going to now go through the lab, because lab reinforces lecture. I'm going to go through the lab, and then I will show you the photomicrographs that you may see. Any questions? Please, no one has asked me a question yet. Okay, let's go through the 8 labs. And then I'll summarize. No, and then I will show you the photomicrographs. I'll show you a little bit out on the internet, and then we'll see where we are if there are any questions. Here is lab 1. For this one, again, you'll need to know the terminology. This diagram could be on the test, but I can't ask you 25, 26 questions. So I may ask, what is this nasal? What is this acromial? What is this region? The mentalis muscle is here. The frontalis muscle is here. The buccinator muscle is here. So I mentioned that this all may be new, but as we continue through the semester, it will help you. This is the sternum bone. This is the pectoralis muscle. This is the antecubital region that we sanitize for taking blood often. This is the brachial, and this is the antebrachial before arm. This is the femoral, so it's the femur bone. This is the kneecap, patella bone. This is the crural region, and I'll also get the back figure. So I may ask you about the occipital bone. Occipitalis will be the muscle, the elbow, the olecranon, the lumbar region, or the popliteal. We'll take a popliteal pulse if that patient is prone, which means lying on their stomach. And this is sural, not to be confused with the front of the leg, which is crural. This is the heel bone, the calcaneus. So this is the calcaneal region. So this will be on there with the anatomical or regional terms. Don't forget to be able to use, and I did use contralateral on the opposite side of the body from the structure. So the gallbladder is contralateral to the stomach. Medial, midline, lateral, outside, further from the midline, superior, inferior, anterior, posterior, AVPD, and then proximal, distal. And here, so be able to use those terms. Again, sagittal, it just divides the body into right and left, but I said into equal left and right. That would be mid-sagittal. If mid-sagittal is not there and just sagittal, then I'm going to look for median as a choice, which is the median is the midline. Parasagittal, again, to amputate an arm. Frontal, the big toe. Transverse, a leg. The dorsal body cavity, PD, dorsal, cranial, and vertebral. And then the AV, anterior ventral body cavities, you have your thoracic with the lungs and the heart, pleural and pericardial regions. And then the abdominal pelvic, in my abdomen is the peritoneal. And that's going through pericardial, pleural, pericardial. And here you have either parietal or visceral, pleura, either parietal or visceral pericardium, and the same for peritoneum in the intestines. Here are the four, here are the 9 abdominal pelvic regions: umbilical, epigastric, hypogastric, and then we have left and right, hypochondriac, below the cartilage, lumbar, and then inguinal, or for the pelvic bone, iliac region. And here are the major organs. Before I get there, here are the major 11 organ systems. So I would say this organ system protects underlying tissues. This organ system contains hair and nails, which would be, or this organ system synthesizes vitamin D. It would be the integumentary. This organ system protects underlying organs. This organ system manipulates the environment, allows us to move. This organ system initiates quick action, nervous system. So just look over these organ systems. If I went through like kidneys, I would include that as a major organ in the urinary system. So your lymphatic returns tissues, protects against infection. So it's got the lymphocytes, macrophages, and also removes fluid and returns it. So that's the lymphatic system, and I would expect to see lymphocytes. And so here's the digestive, respiratory. So just look over this for matching the organ systems with the organs or their function. And here are some of the matchings. and examples of some of the use of the terminology. And here are those to help narrow it down. Gallstones, where's the gallbladder? Hepatitis, which refers to liver, where's the liver? Stomach ulcer, where's the stomach? Intestines are really in all four for sure, and in all of the 9 except for where the bladder is, which is just the hypogastric. The bladder is the hypogastric. Then you've got appendicitis, often seen, right inguinal for the patient. So just these terms to know those organs would help. That's lab exercise 1. Lab exercise 2. That one was on the buffers, adding acid to buffer. Again, I would simply ask for questions. I would ask what happened when acid was added to the buffer, what happened when acid was added to, I'm not going to ask how many drops. I would ask what happened when acid was added to water, what happened when acid was added to buffer, drastic decrease in water, slight decrease in buffer. The buffer, CO2 was released, so we saw fizzing. That's it for lab exercise 2. Lab exercise 3 was a reinforcement of the chemistry, which was catabolism and anabolism. Catabolism breakdown, anabolism, catabolism breakdown, which is hydrolysis, anabolism, dehydration synthesis. And it could be with the carbs, proteins, water release synthesis, water consumed, hydrolysis, catabolism, synthesis. That's lab and there were no questions. Lab exercise 3. Amino acids, lipids, anything, that's how we make or break down. Lab exercise 4. Introduction to the microscope, which we will do. It is a parfocal microscope. Once it's in focus at low power, it will be in focus at all subsequent powers with fine focus, fine tuning. And we'll go through that tomorrow. Your magnification is the object. It is, the image is inverted, which is, so be sure to look at the E on the stage and then look through the ocular, you'll see it's inverted. The way we calculate magnification is the ocular, which is always 10, times the objective, either scanning, low power, or high power. And this is the working distance. I'll go through this again tomorrow. As magnification increases, working distance decreases. And I broke my share of slides, especially with a doctorate in micro. But we're now teaching, start with scanning and then slowly increase the power. And some of these you will see on there. We'll go through these answers tomorrow. I get my questions directly from the lab. That's lab 4. Lab 5, we will do tomorrow. And for this lab, the cell cycle. A short answer would be name it would be name the, okay, here is prokaryote and eukaryote. Prokaryote has no membrane-bound organelles. Eukaryotes do. Here's a nucleus, mitochondria, Golgi. So prokaryote, no organelles, no membrane-bound organelles. Eukaryote does. And here's the cell. There'll be a picture of the cell. Don't forget the nucleolus, where RNA is made. The nucleus, where DNA is housed. The rough ER is right outside the nucleus. It's the site for protein synthesis. The smooth ER, storage for some of the lipids, hormones, etc. Mitochondria, DNA, ATP. Mitochondria, ATP is produced. DNA is stored in the nucleus. Mitochondria, ATP is produced. Lysosomes lyse, peroxisomes oxidize. I won't ask you the microtubules, microfilaments, centrioles, any of these cytoskeletal elements. And there will just be a picture. This is a good review. We'll do this lab tomorrow, where you can practice. Okay, what is this? What is this? What is this? What is this? And cell cycle. Good short answer question. What is the cell cycle? Where does the cell spend most of its life? The cell spends most of its life in interphase. It is growing, G1. It's getting ready to divide, so it's going to duplicate the DNA, and it's going to continue growing and duplicate the organelles ready for division. Now it will go through mitosis, the M phase, which is division, prophase, metaphase, anaphase, telophase. And then it will cytokinase, break into 2 distinct cells. We will go through this tomorrow. We talked about the 3 types of RNA. If I give you one on the exam, this is telophase. I would simply choose, as I said, either metaphase, they are lined up directly on the equator, or anaphase, they're beginning to be pulled apart. And so we'll go through this tomorrow. And here's the good question. What is the sequence of the periods in the cell cycle? Describe the cell cycle. So we'll go through this one tomorrow, lab 5. Lab 6, we did already. And I will take some of my answers, some of my questions from the questions on lab 6. I asked you to, we talked about solid in a solid. So the solvent is the agar. The crystals, a solid also is a solute. And the smaller molecular weight diffuse the fastest. We talked about hypotonic, hypertonic, and isotonic. Hypertonic crenates and hypotonic burst, or we call it, because red blood cells rupture, it hemolyzes. This was a selectively permeable membrane where water went inside the sac, and some of the small molecular weight substrates, solutes, came outside of the bag, trying to equilibrate. So we'll go through these answers. We already went through them. I would study these answers. They're great for the exam. A solution that is hypertonic to another solution means hypertonic means it has more solutes than the other solution. So more solutes in A than in B, because A is hypertonic to B. This is the one I said think about. I'm going to explain it, then I'll explain it again in lecture tomorrow. You have 2 sacs with 15% glucose in one and sucrose in the other. As long as it's not a polymer, it doesn't matter if it's a monosaccharide or a disaccharide. So 15% sugar in each sac. And then each sac is placed in a beaker with 45% glucose, sugar. So if that 45% glucose is in the beaker, that means 55% of that beaker is water. But in the sac, I only have 15% glucose. So that means 85% of that inside the sac is water. So water will leave the sac and go to the beaker. And that's why this is the answer. One of my students finally said the way she understood it was to calculate which one has the most water. Well, we'll go through these. And again, I will take some of my questions from the lab. Now you have lab exercise 7, which is, this is going to be lots of schoolwork after lecture tomorrow. Lab 7 is epithelial and connective tissues. And I'm going to go through some photomicrographs in a minute. Again, it is all epithelial and connective tissue. All you're doing is drawing. The only one I may ask is this one. Yes, I'll probably do some of these. Remember, pseudostratified is not really stratified. It appears to be several layers. So look at these. We'll go through them tomorrow. adipocytes, adipose, chondrocytes for hypochondriac region, chondrocytes or cartilage. We'll go through these tomorrow. And smoker's hack, I like asking this one. Because if a person who sometimes when people smoke, they tend to cough a lot, and that's because the cilia that's on the pseudostratified ciliated columnar epithelial cells in the trachea, sometimes that cilia gets sheared off. It's not spreading the mucus, so sometimes things do get deep in the lungs, and they're coughing to expel that. And then tendons and ligaments. I'll explain that more in chapter 6, but I will tell you that it takes them a long time to heal because unlike bone, they don't have a direct blood supply. And we'll go through that tomorrow with tissues. The last one we'll go through with tissues is the muscle and nerve, and that is lab exercise 8. Muscle and nerve. So let me show you photomicrographs. The one question on this one that I like to answer is when the heart is damaged, when you have a heart attack, the heart muscle, cardiac muscle, is replaced with connective tissue. So you'll hear loved ones say, the doctor said I've had a heart attack. I've had 2 or 3. They can tell because that heart loses its strength to pump because that cardiac muscle is replaced with connective tissue, scar tissue. And then the reason why old people are cold all the time is because we lose the adipose, we have cushioning, and our epithelial tissue gets thinner. So here is did I go through? Oh yeah, I did lab 4. So here are some of the photomicrographs. Remember we said we will have this is a culmination of all of the labs for module 1. So I just went through lab 1 with you, lab 2, lab 3, lab 4 we'll do tomorrow, lab 5 we'll do tomorrow, where you'll look at slides of mitosis to recognize metaphase and anaphase. And those are the 2 I kind of stick to. A short answer, describe prokaryotes versus eukaryotes. There'll be a cell there, and I'll say name this structure, name this structure, name this structure. And just the ones that I pointed out. Don't worry about the microfilaments or any of the cytoskeletal elements. I won't ask those. So if I give you mitosis, it will either be metaphase or anaphase. This cell is in metaphase. The chromosomes are right down the middle on the equator. This is anaphase. They're beginning to be pulled apart. Telophase is kind of beginning to invaginate and go through cytokinesis, but I want you to at least get these that are pretty definitive. Metaphase and anaphase. These are the 2 phases of mitosis I would ask. Here it is in a different view, anaphase and metaphase. Telophase, they're beginning to invaginate, but I'm just telling you, I'm going to skip with metaphase and anaphase, beginning to be pulled apart directly on the equator. Then lab exercise 6, we went through that. And I talked about, this is what I wanted to tell you. So what I will do is put a box. I think you can see my arrow. I will put a box over A, B, and C, and I'll just leave the A, B, and C there. And I will say, what type of solution were the cells in the image label B placed in? Well, if that cell is shrinking, it was a hypertonic solution. If the cell is swelling, lots of water is going in and it's about to burst, that would be a hypotonic solution. And then isotonic, the red blood cells look like themselves, they don't change. So the question, that's why I said no isotonic, hypertonic, and hypotonic. So I would just use a picture like this, cover the words and say, what type of solution are the cells in A, B, or C placed in? So here are the tissues. I said, name the 8 types of epithelial tissue. It is either simple or stratified, 1 layer or more than 1 layer. That's the first word. The second word is what's the shape of the cell. So it is either simple squamous, cuboidal, or columnar, that's 3, or stratified, squamous, cuboidal, and columnar, that makes 6. Number 7 is transitional, and number 8 is pseudostratified. So here I'll have this photomicrograph. Name this tissue type. Simple squamous, flat, 1 layer, simple squamous. And it's in the alveoli. Remember, click gas exchange. This is the kidney tubules, and it is simple cuboidal. They're not perfect cubes, but they look square. So, and it's 1 layer. So this is simple cuboidal. In the kidneys, a little bit more time to remove the waste, filter the waste. Kidney tubules. There are other areas, but I'm trying to give an obvious one as you learn where to find these tissue types. Simple columnar in the intestines. I need more time to absorb all of the nutrients. Also, when you get to that organ system in AMP2, they're going to talk about microvilli, or the brush border. I got all these nutrients that I need to absorb in the simple columnar, and rather than going like this, how about if I have finger-like projections? Oh, now I have all this surface. I have all this surface area to absorb nutrients through, and that's what these microvilli are, finger-like projections. Notice the difference here with my simple columnar in the intestines and the goblet cells to add mucus to make the removal of waste easy. This is a simple columnar with the microvilli. Here is the pseudo-stratified, ciliated. This is hair, different from the microvilli. Pseudo-stratified, ciliated columnar epithelium. Here are my mucus cells, because I said they're going to spread mucus in the trachea, trapping dirt. And here is an artist's rendition. Yeah, they look like they're stratified, but it's just 1 cell. The nucleus is located in a different area. So be able to recognize this on a photomicrograph. Here is stratified squamous. When we get to chapter 5, I'll talk about the top layer of the skin sloughing off when we shower. You can see it. So this is stratified squamous. Stratified squamous, the skin. Other areas, but stratified squamous. I'm not going to have a picture, a photomicrograph of stratified cuboidal, but I want you to know it's one of the 8 types. I'm not going to have one of stratified columnar, but I want you to know it's one of the 8 types. And here's transitional, the 8th type. Remember, I said it is the bladder, and it looks like it's transitioning. Here it looks columnar, then it looks flat. It just depends on the extent of the bladder. So if it's a full bladder or not. So this is transitional. Here is loose areolar connective tissue. It's in the various areas of the body. It's actually in the papillary layer, but we'll get to that with chapter 5. This is adipose. I want to show you one out on the website, because I didn't use this photomicrograph, but the bottom line is it's a big vacuole. Some students say cotton candy. It's a big vacuole. This is adipocytes adipose tissue. I didn't have this one on there, reticular. The purpose of this in the spleen is to remove dead red blood cells. So it's like a mesh, but this is reticular. I don't have that one on there. I don't have dense regular connective tissue in tendons and ligaments. I do have this one. It is in the aorta. It is elastic connective tissue, not to be confused with the elastic cartilage. Elastic connective tissue in the aorta. So as the blood pumps from the heart, the aorta expands and contracts, expands and contracts. Some students say beef jerky, but it looks like an elastic waistband, elastic connective tissue. I don't have this one on there. Dense, irregular. It is in the 2nd layer of the skin, but I don't have it on there at the photomicrograph. I do have this one. Tomorrow, when you look at your slides, I promise you, they look like this. You're going to try to simulate the same. So 40 is high power, but sometimes I go low power to look at it. But this is textbook perfect. This is hyaline cartilage. It will be on there. I will also have this one on there, fibrocartilage, because it looks like that, right? still have your chondrocytes inside lacunae. This is fibrocartilage. Remember, in the knee, in the vertebrae, and in the pelvis. I will not use elastic cartilage. That's why I want you to say elastic connective tissue for the connective tissue. Here's bone. Looks like a tree with tree rings. Bone. We're going to go through bone. That's one of our organ systems. We'll talk about it. But tomorrow, I usually go low or scanning. It will look like this. I promise you. So I want you to be able to recognize bone, a connective tissue. Blood, a connective tissue. White blood cells, lymphocytes, red blood cells, platelets. It is a liquid connective tissue. And nervous tissue. Here's the neuron with axons and dendrites. Nervous tissue. It may not be this type of stain. It may be purple, but you will still see the neuron with axons and dendrites. The fourth type of tissue, third type nervous tissue. We talked epithelial, connective, nervous. Here's the last one, muscles. Practice, practice, practice tomorrow. Our muscles slides don't look like this. So the picture I put on there doesn't look like this, but definitive striations versus faint striations. The skeletal muscle has definitive striations and it is multinucleate. Practice tomorrow. Multinucleate definitive striations. In the cardiac, you see these intercalated disc lines. They connect cells to cells. That's not in this one. So you see intercalated disc in cardiac, the striations are a little fainter. And it's not multinucleate. And then this is smooth muscle. A lot of my students mistake this for epithelial tissue or other tissue. They are spindle-like cells. Some are spot on. It's smooth muscle. It's in the organs we don't control. But if you look closely, you can see the spindle-shaped cells. Spindle-shaped cells, smooth muscle. So the three types of muscle you'll need to be able to tell those apart. So we just went through all of this. These are all of the ones that I said. Stratified squamous, simple cuboidal, simple columnar, bone, blood, pseudostratified, transitional, nerve, and for the connective tissues, adipose, areolar, loose areolar, elastic connective tissue. For the cartilage, hyaline and fibrocartilage, and for the muscles, all three. I wanted to show you one more thing out on the internet. For the adipose, it may look like this one. You can tell they're the big vacuoles, so you can tell that this is connective tissue. This one is closer to ours, but if I gave you that one, it's connective tissue. I'm sorry, don't tell me connective tissue, tell me specific adipose connective tissue. If I were to use this one, you see a lot of vacuoles. So questions for me. I kept you way longer than I wanted to, but that's because we haven't covered a lot of it. So tomorrow should be redundancy. And the more we talk